Hi, everyone. My name is Randy Oliver. I'm a professional beekeeper and bee researcher in California. I run the website scientificbeekeeping.com. Apologies for having to record this in advance. I hope you enjoy the presentation. One thing that comes with experience in beekeeping is being able to make your management decisions based upon what you actually see within the hive. I call this reading the combs. The problem we have these days is there's so much conflicting advice on the internet about beekeeping. It can drive you crazy. Now, the problem is a lot of it, that advice is put out there by people who really don't have that much experience with beekeeping. And they would do well to read this paper by Denning and Kruger about how you can fail to recognize your own incompetence and have inflated self-assessments. And what I see, this happens with beekeepers. You start with your first hive, and this is your degree of self-assessment of your knowledge and ability. And this is your years of experience in beekeeping. And you start off, and boy, your first year you do really well. You think, oh man, I'm smarter than every other beekeeper who ever existed. In the thousands of years people have been keeping bees. And you start your blog on this. And then the next year, Varroa catches up and all your colonies crash. You reach the trough of disillusionment. And some people give up at that point. But then a lot of them get back on the horse, get some bees again, and start on this slope of experience and enlightenment, which takes years and years, and viewing thousands of combs and colony inspections as you slowly gain mastery of the craft. And the beauty is you'll never understand it all. There's always going to be questions. After 50 plus years of beekeeping, I have more questions now than I started with. So keep this in mind. So instead of listening to other people, here's the rules of beekeeping from the bee's point of view. And this is the big shift, is to see the world through the bee's eyes rather than human eyes. The first rule is they need a warm, dry cavity. They typically find that in a tree cavity, or the beekeeper can provide a cavity, anything. The bees don't care how it looks, what color you paint it, or whether it's a top bar, or a Warri hive, or a Langstroth hive. They just need a warm, dry cavity. Number two, they need food. And their food is pollen or nectar. And if food is unavailable, you can either move your colonies to a better location where food is available, or you can step up, do some husbandry, feed them some sugar syrup or other form of sugar to provide carbohydrates, or a pollen substitute or pollen sub for protein. The third thing is that every species has parasites, and the honeybee is in the middle of this evolutionary adaptation to a novel parasite, the Varroa mite. And the Varroa mite has just wreaked havoc because it transmits viruses, especially deformed wing virus. And during this evolutionary process, to be a successful beekeeper, unless you are lucky enough to have mite-resistant bees, and I do have a proportion of those in my operation, you'll need to control this parasite in order for your colonies to thrive. And that's the end of rules. Anything else anybody says is a reflection of that person rather than what the bees need. They need a dry cavity, they need food, and they need their parasites controlled. I have a simple recipe here for keeping healthy hives. Young, vigorous queens. In nature, typically a queen runs out of sperm after about 18 months of egg laying. And so requeening colonies each year gives you more robust colonies. Good nutrition, either by good location or by supplemental feeding. Control Varroa all year, not just once or twice. You start early in the spring and all year control. Minimize the toxins. That's both agricultural chemicals, toxic plant nectar and pollen, or anything that the beekeeper dumps into the hive, like essential oils. Many of those are irritating or harmful to the bees. And the number one thing is to be proactive rather than reactive. Many beekeepers do everything reactively to a problem, where professional beekeepers do things proactively before the problem occurs. I do have some suggestions. First is that honeybees are a fascinating animal, but they're just animals. And keeping them, think of them as livestock and practice basic animal husbandry. It all applies. Number two, copy consistently successful local beekeepers. Now, consistently successful doesn't mean the ones who make the front pages of the paper or talk the loudest or are the leaders of your meetings. Success is the sign of success for a beekeeper is that your problem in the spring is you have too many bees. And if you don't sell some or give some away, they're all going to swarm. If a beekeeper is not able to, if that beekeeper's problem is not having too many bees every spring, I would not consider them to be consistently successful. Suggestion number 3, understand the 4 horsemen of bee apocalypse. Bees are a tropical organism, has adapted to living in colder climates. So chill is a big problem for honeybees. Poor nutrition, huge problem, as with any other species. Here in California, we deal with this all throughout the late summer when there's no natural pollen coming in. Toxins, of course, and it's not just pesticides. It's what the beekeepers put in the hives, these snake oils and stuff, or natural plant toxins. We have toxic plants here in California. And the last, parasites, notably varroa, but also European foul brood, American foul brood, nosema, and viruses are parasites. My 4th suggestion, base all your management decisions not on what somebody tells you to do, not a cookbook recipe, but upon the biological needs of the colony, and for you to understand those biological needs. And to that end, I'm going to, the rest of this PowerPoint will be about you learning how to read the cones and determine those biological needs. Here's a quote that I like a lot. It's by Ticknor Edwards back in 1920. The bees have their definite plan for life, perfected through countless ages, and nothing you can do will ever turn them from it. You can delay their work, or you can even thwart it altogether. But no one has ever succeeded in changing a single principle in bee life. Let that sink in. No one has ever succeeded in changing a single principle of bee life. And so the best bee master is always the one who most exactly obeys the orders from the hive. It's not so much the orders from the hive, but learning to see what the hive needs by looking at the combs. One thing I want to talk about is understanding the niche, the ecological niche of the honeybee. And a niche is simply a business model that an organism has. The honeybee has customers for their business, and those are flowers needing pollen transfer, and the flowers reward them for doing that job. The honeybees also have competitors. All other pollinators and any other beehive is a competitor. Plus, they have parasites and predators, such as birds and stuff. Success is all about how well the individual colony bee can exploit the available resources at any time, and doing so competing against all the other colonies in the neighborhood or the apiary and other wild pollinators. And then the winners get to pass their genes on to the next generation. This is how it works in nature with the bees. So understand that the honeybee organism is this highly efficient competitor, okay, for those specific and limited floral resources at any time of year. The objective of the honeybee colony, our objectives are 2. One is to store enough food. So here's the honey, the food storage, so that the honeybees can maintain a live, heated colony during dearth, especially during winter and cold weather that no other insect can do. And then, when times are good, to send out drones and swarms to propagate its genetics. Those are the only 2 objectives to the colony. The next thing is that it has a competitive advantage because it maintains a standing army of foragers. So it competes against other pollinators, other bee species who start with a single female and they raise their offspring one at a time. The honeybee maintains a standing army all the time by keeping that honey reserve to keep them alive. And that standing army of foragers, they don't fly. They don't do any work unless the scouts come back and tell them there's something out there. So until the scouts go out in the morning and come back, the rest of the foragers just sit and hang out and save their energy in the hive. They don't waste any unnecessary energy. Everybody's aware of the queen, this unusual large insect. There's nothing really unusual about a queen bee because she's simply a viable female reproductive insect, like any other female reproductive insect. The amazing thing with the honeybee colonies are all these workers who are non-reproductive females. That's what makes honeybee special, colony special, not the fertile queen, but all the non-reproductive females that do all the work in the colony. So the queen functions as the ovary and the pheromonal center of the colony, but she has very little other influence in the colony. It's the nurse bees who actually run the show, and they regulate colony growth or colony decline by controlling the food supply, producing and controlling the supply of jelly that they then transfer to the larvae and to every other bee in the hive, including the queen. That's the source of food. So the nurses are the source of food for the colony. The other thing is that all the members in the hive, by pheromones, are aware of whether there's brood being reared or not, whether the queen is present. They scout out the storage capacity, amount of empty comb or combs full of honey in the hive, and they all share food or starve equally. So here's a colony in the middle of starving out, sharing food. And all but the queen are considered to be expendable, including all the brood that they cannibalized for protein supplies because they've run out of food reserves here. So they all work together, live together, and or die together. The most important thing is to understand to read the cues in the colony. And the nurse bees who respond to the cues from the brood, their pheromones, and from what the foragers are bringing into the colony at any time. To understand the dynamics of the hive population over the course of the year, it's all about recruitment versus attrition. With recruitment is the rearing of new worker bees, the brood. And this is the main thing that the colony does to deal with problems, with disease, with starvation, with toxins. They simply rear a bunch of new bees to take the place of the old ones. There's a very rapid turnaround of the population during the spring and summer. Attrition is the bees flying off to die. And this point of flying off, very rarely does a bee die within the hive. They have a very strong instinct to remove their bodies from the hive when they start to feel ill or too aged and die outside of the hive, so the sisters don't have to deal with their dead corpses. And the growth of a colony or the decline is simply the mathematical relationship of recruitment versus attrition. So this is the daily gain in total number of bees in the hive here. So during rapid spring buildup, a colony builds up, adds about 400 to 600 additional bees to the colony as a whole every day. Now, if the queen's laying 1,500 eggs a day, that means that 1,000 bees are dying of old age or other attrition every single day. So this very rapid turnover here. Finally, the colony comes into equilibrium with the death rate, the attrition of bees, and the recruitment rate. And that works out to be about 40 times the egg laying capacity of a queen. So if a queen's laying 1,000 eggs a day, the colony is going to grow to about 40,000 bees. If she's laying 1,200 eggs a day, they may get up to 50,000 bees. And then when the queen slows down on egg laying, then you have this attrition exceeding recruitment, and the colony population will drop back down again. So this is a 2-year curve of colony population. Number of bees here, and dates blanked out because dates mean nothing. The bees respond to the availability of pollen and nectar in the environment. So in the northern regions, we think it has to do with the winter solstice, but if you move south, it has nothing to do with day length or the solstice. I've been in Australia where during the winter, on the winter solstice, the white box comes into bloom, and those colonies are rearing brood and building up and storing honey like crazy on the shortest day of the year. Down in Mexico, when I visited the beekeepers there, their winter, where the colony population is at its lowest, occurs in July and August. We have a similar situation here in California where it dries up because there's no pollen and nectar coming in. So forget that it has to do with month or day length. It has to do with pollen and nectar availability. And there's a lag in population just a few weeks behind the amount of incoming pollen and nectar. And the colony goes through 4 phases a year. When pollen becomes available, protein's available, it starts the buildup phase. When it gets about halfway built up to maximum size, reached its maximum brood capacity, it goes into reproduction, which is swarming for the bees. And then it goes into the food storage phase during the main honey flow. And the honeybees are all about this. If they don't store enough food during the main honey flow, the colony will not make it through the winter. So that's really important to them to store enough food. And then afterwards, they go to the their survival stage. They reduce the number of mouths to feed, and and then this hold out until they go back into the buildup phase. Okay. There's a graph I created from some data by Lloyd Harris from Manitoba, Canada. I'll refer back to this, and I have a series of about 13 or 14 articles on my website that goes over how to interpret this throughout the course of the year. I highly recommend you look at those. So here's the colony age structure. What I did is I put the numbers over the months, Northern Hemisphere, January through December, the amount of brood, this dotted line, so this would be eggs, larvae, and pupae in the colony at any time, the total amount of adult bees, which you can see here. So at this time of year, you have twice as much brood as you have adult bees. And then the age distribution of the bees. These are red as bees that are 0 to 12 days of age, the adult bees, 13 to 24, 25, 36, and very few bees make it past 48 days during the summer. Then we have the spring turn over here and the fall turn over here. And the winter bees or diutinous bees are these blue and purple colored. These would be summer bees here. And the colony goes through these different biological phases throughout the course of the year. And that's what my series of articles covered, an article typically for each one of these phases for deeper understanding. Another point I'd like to make is I suggest you don't do anything in the beehive unless you first got a plan of action. Don't open the hive until you know exactly what you're going to be looking for and exactly what you're going to do if you find something. So think first before you open hives. Otherwise, don't bother the bees. Other than varroa management, the bees are generally better off without you going into the hive. With varroa management, we need to, but understand why you're going to be doing something. Don't just go in the hive just without knowing what your plan is. Understand your local progression of bloom, the phenology of the plants, and every bee club should make a chart like this for their members with the dates over the year. But more important than the dates is what is blooming. So here where I live, we have apiaries within 20 minutes drive of home that are a full month different between when things bloom. So we can actually get our entire blackberry flow, honey, from one yard and then pick those bees up and move to the beginning of blackberry flow in another yard less than 30 minutes away because we have an elevation change. So make the chart with phenologically based. Put in, show the colony population when you typically swarm, when you have toxic plants like we have buckeye here, when you have your dearth, and when you have your main honey flow. And then what you'll notice from the time brood rearing is initiated until swarming is typically about 60 to 90 days. And then to reach colony peak population again, it's about 30 to 60 days. So you do everything, all your management, working backwards from these known time periods. So to avoid swarming, you want to count backwards to avoid this. To build your colonies up for main honey flow, you want to understand this figure here. Here would be a typical management. This would be from my area in the California foothills. When you're going to be looking for different pathogens, when to be proactive with varroa, when you may need to do supplemental feeding for the bees here, when to check for adequate stores. Your time for buildup right here, when to look out for starvation. A little chart like this for your local club will really help your members. Then they can count backwards for their management decisions. Now, as far as reading the combs, you want to be able to recognize certain things. Whether there's fresh pollen coming in, whether there's fermented pollen or bee bread, which is stored for later use. The nurses, of course, prefer fresh pollen over stored bee bread. They will consume the fresh stuff first. If you see bee bread, that means there's been a surplus, more than they need to eat. You have honey, so this is the dark cappings. This is the older honey because the wax has lost the airspace and is touching the honey, so it darkens it up, darkens the look of it. You have the nurse bees, which are the ones that consume the bee bread here. You have the open brood putting off pheromones, very hungry for food, and the sealed brood, which is also called varroa food. This is where varroa reproduces here. So the question is, if the nurse bees are running the show inside the hive, how do they know what's happening on the outside? So I'm going to give you a view of the inside of the hive from the nurse bee's perspective. It's dark in the hive. They can't see a thing. Everything is done by odor and taste and touch inside. So forget being a human. We are dependent upon our vision. Inside the hive, the bees don't use their vision at all. What the nurses do is they monitor the amount of honey reserves to see whether it's worthwhile for them to, whether they can afford to start rearing the brood and making a brood, heating up a brood nest. Whether there's incoming nectar, that's a sign, wow, this is the time for us to start to build up more brood here. If there's fresh pollen coming in and if it's abundant, and then with those larval pheromones, this larvae give off a pheromone called E-beta osmine, which is critical. That's a signal to all the bees in the hive that they have a queen who is laying eggs and that the nurses are feeding those larvae. So that tells every other bee in the hive what's happening. If that E-beta osmine signal fails, that tells them they need to rear a replacement queen right away because the queen is failing, or they need to make swarm cells because they've run out of room. So this larval pheromone is very important. And the interface to pay attention to is between the honey and the brood. This is the dynamic interface that you want to watch. The honey is the energy reserves, the stored honey, okay? And the bee bread is any protein reserve, other than also the brood is a protein reserve because it will be cannibalized if they need it. The brood's also, the open brood is the energy and protein demand in the colony. Then there's two interfaces. You have the energy interface, which is usually a single line of cells right between the honey and the bee bread. If this is freshly opened honey and ripened honey, then they are eating their way up into the interface, into their stores. If it's nectar coming in, being processed in the honey, then they'll be pushing their way down in the pushing the brood nest down as they expand their reserves. The protein interface is right here. The foragers come back and they fill the cells all through the brood area. Any empty cell, they'll put pollen in here. You don't see it there normally because the nurses preferentially consume the pollen right here. The only pollen they don't consume is which is out at the edge. So we see this nice band of pollen, but that's not the way the bees put it there. That simply means there's pollen left over that they didn't consume from this area. And if I'm doing a nosema test where I'm crushing bees in water to do microscopic inspection, if I took a sample of nurse bees, that water would be stained with the yellow in the nurse bees' guts. Whereas the foragers don't hold pollen in their guts, they don't digest pollen, and that will look clear here. So the nurse bees are the only ones in the hive that do this digestion of the pollen. The rest of the bees, the pollen passes pretty much undigested through the gut. Those nurse bees then are the stomach of the hive. They're the ones that utilize that pollen coming in from the foragers. And that can't be used directly by any other bees, by the brood or the queens, not at all. The brood can digest pollen, but doesn't need any pollen. And the other workers beg the nurses for a feeding of protein in the form of jelly, this trophallaxis, this transfer. And this communal stomach of the bees is this shared currency that lets every bee in the hive understand the nutritional status of the hive. So when a forager comes back with a load of nectar, unloads that load of nectar and says, wow, I need some protein to rebuild my flight muscles and for maintenance, they don't eat pollen. They go to a nurse and they try to beg some jelly. And the nurse says, oh, sorry, dude, we don't have enough protein coming in to give you this jelly. Well, that tells the forager, wow, I better go forage for pollen instead of for nectar. So you have this feedback loop that regulates the foraging. So let's start here at the buildup phase of the colony. The first cue in the springtime are the first tree pollens coming in. Where I live, it's alder trees. Other areas, it's different trees. And these are generally wind pollinated plants, but some of them have very high nutrition. Our alder has very good nutrition for the bees. This is the cue that triggers the buildup of the brood nest. You can see here from Manitoba, starting in March, the first tree pollens came in, and notice, bingo, up goes the amount of brood rain right here. Here's a picture from my operation in California. Here's fresh alder pollen right here, brought in, and the bees are initiating brood break. This happens in the second week of January, where I live. And that fresh pollen comes in, stimulates the nurses to start producing a bunch of jelly, and they will stimulate the queen to start laying eggs by feeding her more jelly, and then um, uh, they will feed all those larvae because there's plenty of jelly coming in. The pheromonal signal from those larvae then stimulates the nurses to really start eating a lot of pollen. They get voracious for pollen and producing a lot of jelly. It also is a signal to the foragers that we have hungry larvae. We should preferentially feed forage for pollen. And then we get to the spring turnover. And this happens because the number of adult bees in the hive starts to decline as they age, and while the amount of brood starts to increase. And we can see this happening right here. You have these long-lived winter bees, or diutinous bees, that are almost immortal. Look at this, almost no decrease in their population for months there in Canada until they start rearing brood. The moment they start rearing brood, then they change their physiology from long-lived bees to summer bees. And as soon as this brood starts to emerge, they then shift from these nursing duties to mid-age and then foraging duties, and then die fairly quickly after they wear out from foraging. So look at the sudden decline of the adult population that occurs about 30, 35 days after they initiate brood rearing. Meanwhile, you get this increase now, this recruitment of these young bees that need to take the place of the older bees. So this is called the spring turnover, and this is often the most critical time of the year for a colony in cold winter areas, because if the weather screws up here, these bees die off without enough bees recruiting coming in to take their places. And notice the demand on these nurse bees. There's maybe twice as many immature bees in the hive as larvae or pupae as there are of adult bees to care for them. In areas where there's a big, strong early spring bloom, such as almonds, which occurs in middle of February during winter, the bees respond to this big bloom and start foraging like crazy. And when they start foraging, they wear right out. They only got about 8 to 12 days for an adult bee before it wears out when it's foraging. So you can have these colonies collapse very quickly unless they already had a large brood nest of emerging brood at the time when the bloom starts. Here's a picture from late January I took of a colony. Notice here's the honey reserves. Almost no pollen reserves, but look at this little bit of pollen up here. Look at all this beautiful brood right here. So there are barely enough bees to keep this brood warm and fed. So they're desperately rearing their replacements. And then this is when we start getting problems with the parasites or pathogens. They're all parasites, but they're seasonal. This is over 2 years again. You can see during the winter, nosema tends to be the problem, especially in the springtime. European foulbrood and shockbrood also in the cooler weather of spring. And then these kind of disappear. During the summer, nosema is not normally a problem. Possibly American foulbrood during the summer. The Varroa deformed wing virus become a more serious issue in the late season, late summer to fall. And if you have paralytic viruses and nosema at the same time, that can cause colonies to collapse. There's a picture of a bee's gut I took 400 times my medication. Here's the tracheal tube. You can see the striations of the gut tissue here. And all these little tiny white ovals, each one of those is a nosema spore. And you can see how the nosema just fills the gut tissue full of spores. And it uses up the energy and the protein of these gut cells, and also suppresses immunity, suppresses apoptosis of these cells, which is the way the bees fight viruses. So nosema can be quite stressful to the bees. Here's a 3-frame colony on the verge of recovery. But if you take a look at this, This is taken in January when it's cool at night. And you could say there's not enough adult bees to keep all that brood warm on a warm night. This colony that attrition is exceeding recruitment, and all it will take is one really chilling night for this colony to be in serious problem. And you'll see a couple of days later, you had a cold night, this cluster contracted, the brood got chilled. You can see the ring of bee bread where that tells you this was all sealed brood all the way out to here. If I take these bees and look at them one at a time under the scope, I'll have a very high percentage of them infected with nosema. Typically 8 to 10 out of 10 bees, 80 to 100% of these bees will be infected with nosema. Whereas a lower rate of infection, the colony would not have collapsed here. Here's another typical nosema collapse. This is what we saw during the invasion of Nosema ceranae back in the early 2000s. The silver dollar cluster of bees left with the ring of brood unattended around them. We don't see this much anymore. Many beekeepers are confused as far as dysentery. Nosema, there's no evidence that nosema causes dysentery. This dysentery is a problem of too much moisture in the bees' guts, and they're unable to defecate in the hive. So when they have a warm enough weather, they don't wait when they get out and they let it go, and that causes this dysentery. If they do defecate in the hive, dysentery can spread nosema spores, but it's not an indication that there's nosema. It's just indication that nosema could spread. So don't get dysentery and nosema infection confused. The only way you can tell if a colony has nosema is with a microscope. And here's a picture right here. This is at 400x, but enlarged for this photograph. Picture of a bee's hair for reference, or a pollen grain. These are pollen grains for reference. And these would be spores of Nosema ceranae here. They're a little more elongated than Nosema apis. And here's the thing, Nosema usually does not have that much effect upon a well-nourished, strong colony with a young queen. But if it is prevalent, it can slow down, hamper the turnover of the colony in the spring, and slow down colony buildup and honey production. Okay, if they get past the spring turnover, they go into the linear growth phase. And if there's plenty of pollen and nectar coming in, they can grow very, very rapidly. Here's data from Nolan, 1925, showing this linear growth of these colonies until they come into equilibrium with the queen's egg laying rate at about 42 times the egg laying rate right here. And we can see it here with Lloyd's data, this linear growth of the colony population. Now, when they're growing like that, they're using a lot of energy to heat up the brood nest and to feed the brood. And they can be living hand to mouth. So here's colonies that singles that we have built up strong. We're putting a second brood chamber on. But this, if you heft this colony, it doesn't have much weight. And this colony can just starve overnight very easily. During that spring buildup in summertime, there's a complete turnover of the worker population roughly every month. That requires, just for the protein to replace those workers, about a pound or two of pollen coming into the hive every single week. Just understand how important this protein coming in is. Notice here's a colony growing up, and they're eating all their pollen every day. This little tiny bit that's left at the end of the day, by morning, there may be none. So you could open a colony early in the morning that's rapidly building up in the springtime and not see any pollen reserves at all. They're living hand to mouth. That's typical. And then if it rains, with Carl Krelsheim found that even within an hour of rain starting, the nurses start to cut back the amount of jelly that they're feeding to the larvae. The colony goes back into survival mode. You can see here in Lloyd's data, there was a hiccup in the amount of brood. And when I checked back with the weather history, I found out, yeah, there was a cold storm moved in on this week when he collected this data right here. And you'll often see that the bees will cut back on brood rearing. If it's a little more intense, and they start to run out of stores, as this colony did. We had a freak storm, a snowstorm in May a few years ago, and many of our colonies consumed all their stores and then consumed their brood, cannibalized their brood. They don't normally cannibalize the sealed brood. They will cannibalize any open brood that requires feeding. So this colony is in desperate survival mode. And here I am years ago treating my bees better than I treated my own children, because I say, boys, time to put on the weather gear, foul weather gear, and let's get out there. And this is the way in California, where we have these rainy springs. We may work bees every day for a month wearing foul weather gear every time we go out, because it's raining or snowing every single day. So we work all day long in this gear. This is beekeeping. Somebody said, Randy, you mean you're out working bees when it's raining and snowing? I said, well, duh. When it's warm and sunny, they feed themselves. Our job as beekeepers is to take care of them when the weather is not conducive for them feeding themselves. We'll also, if they don't have enough reserves of honey, we'll give them some sugar syrup in order to make an artificial brood nest. And everybody asked me about this grass. It was just a marker. We used green grass as a marker when we went through, said, hey, this colony is going to need food. We hefted them. And we know the next visit we come back, if it's dried up, it's not a fresh marker. Hive inspection, checking your mouth. Typically, you only need to pull a single frame out of the center of the upper box in a 2-chamber hive. So you see some honey right here. This tells you have adequate honey reserves. It tells you have good egg laying, high larval survival because you have a solid brood pattern, so you don't have any brood diseases. They got a little bit of bee bed reserves. This is it. That's all the inspection you need other than monitoring for varroa. So here's a nice frame of healthy brood. This is all you need to see to say, hey, this colony's in good shape. Leave them alone. Spotty brood is going to tell you, well, maybe we should look. Maybe we got some European foul brood. Maybe we have nutritional issues. Maybe we have toxic pollen coming in. The other thing to do then is to look at the open brood, the open larvae. Now, a healthy queen lays all the larvae the same age next to each other. If you see older larvae sitting next to younger larvae, that means these larvae are dying for one reason or another. And then the queen comes back, lays another egg, and you get these uneven patterns. This means something is wrong in that hive. You may see chalk brood, a fungus that grows inside the bees, and then the bees chew off the top. You have this very distinctive looking white and tan look. Normally, there's no treatment for chalk brood. It usually goes away, or you can requeen the colony. The one we're seeing a lot of in recent years is European foul brood. And I never used to worry about European foul brood. As soon as you got a good nectar and pollen flow, it would just go away on its own. But nowadays it doesn't. And any colony in the springtime that doesn't build up, if it has a spotty brood pattern, we suspect European foul brood. Here in the US, we can use oxytetracycline to treat for that. Works very well. In other areas, you may have some resistant strains of European foul brood, might be a problem. There's other ways to manage it, but in the US, oxytet works quite well for us. And then it just goes away. We don't see it much at all. We saw a lot of it a number of years ago, and then the bees adapt. This may be due to transgenerational immune priming by the bees, so they become resistant to it. If you see a lot of mixed pollens coming in, you can generally assume they have good nutrition. Just like if you go to the buffet and you bring back a plate with a bunch of different colors, it's probably more nutritious than a plate of all the same color. What we do is we focus on looking for the amount of jelly that the nurses put around the larvae. So a colony with high nutrition, they will cover the cells of the second instar larvae. This is the second instar larva, wall to wall with jelly. And if in this condition, there's nothing you can do wrong. This colony is going to grow like crazy, resist most pathogens and parasites, recover from pesticide kills, recover from anything, because they're recruiting like mad. On the other hand, if the nurses are only feeding enough jelly to those larvae to keep them alive, these will survive. They will emerge as adult bees, but they will be a different adult bee. They're not going to live to their full potential, and they'll start foraging earlier in life and live a shorter adult lifespan. Here's larvae again, very well fed. And again, this is especially during the summer. This is what we look at to see whether our bees need any supplemental protein or not. And here's again, larvae, eggs here, and when they're cutting back the jelly like this, the nurses may just start cannibalizing the eggs as fast as the queen lays them. The queen keeps laying, but the nurses they don't even bother trying to feed them because there's not enough food even to feed these larvae. Okay, so let's just say we're still growing and we get to where the colony tops out in the amount of sealed brood that it has, the amount of brood in the colony. And this is time for the colony to reproduce right here, especially if there's nectar coming in and they start backfilling the honey reserves up to the level of brood. Now, suddenly there's no room for any new eggs, and 3 days later, there's no young larvae and no E beta osmine, no young larval pheromone. And that's a sure trigger for a colony to start queen cells and start swarming. So here's queen cells here. They typically will not start these until they've got drones going already. So this is swarm preparation. And it's natural behavior for any colony when they get the signal or the cues that they filled their cavity, because they don't realize they're in a beehive. They think they're in a cavity in a tree. That's only the beekeeper that realizes that they're in a hive rather than a cavity. So when they feel that they filled the cavity full of honey and brood, the only reason for them to survive, remember their business model, is to then start sending out as many drones and swarms as they possibly can. From that point on, they're just a reproductive unit trying to swarm over and over again until there's free room inside that cavity. And what we do is by managing them, putting empty supers, reversing the brood chambers, we can largely prevent swarming by putting drawn comb above the queen in the brood nest. She'll move up. Now, if you can't suppress swarming that way, and you start to see cell cups, it's too late to try to suppress it. It's time to make splits. Here we are. We set up an assembly line. We make 150, 200 nucleus colonies a day until we've split all of our hive. We make about 3,000 nukes every spring. We work away, big tip, work away from the parent hive. Any bee that's going to sting you flies back to the parent hive. So this person has to wear bee gear. But you can work without gear over here because the only bees that are left here would be the younger bees that are going to stay in these nukes. And what we'll do is we'll put the queen back here at the parent hive. She'll catch the full field force coming back, and this colony will very quickly recover with the old queen. And all the young bees and brood go off on a truck to a new yard. And if you don't manage to do that, you get into colony reproduction here now. Now, at that point, you have a lot of sealed brood in the colony. And sealed brood is stacked like cordwood, very close together. When it emerges, one adult bee covers 3 cells. So you have a 3-to-1 expansion. So a full frame of sealed brood emerges into 3 frames covered with adult bees. This is how the colony very rapidly recovers its population after swarming. And now we get to the main honey flow. The goal of the colony and the goal of the beekeeper is to reach peak population at the start of the main honey flow. If they get peak before, they may starve to death. If they reach peak afterwards, then they're too weak to take advantage of the honey flow and may have too many bees now and may starve to death. So you try to manage your colonies to reach peak population at the beginning of the main honey flow. The other thing is, the stronger the colony, the more efficient it is at putting on honey. So you're better off combining weaker colonies just at the beginning of the main flow than trying to run them as individual weaker colonies. Because the combined colonies will make a whole bunch more honey, and you can split them afterwards. Then the honey flow starts, and there's extra nectar. This is sugar or nectar shake right now. This was early in the morning off of blackberry nectar right here. And they'll put that nectar right into the brood nest, and then the mid-age bees have to move that nectar up and store it as honey above to the sides. And this nectar forces the brood nest downward, and they change everything out. These are honey bees. Storing honey when it's available is more important than anything else. So they shift to making room for honey storage, and they deprioritize brood rearing. And if they run out of cells where they can store that nectar, that's the cue to trigger production of beeswax. So you'll see this white wax right here. White wax means the colony is running out of room. You need to give them more room, or they will swarm. It also is the only time that they will draw foundation. So there's no tricks for drawing foundation. If the bees are not producing wet wax, they're not going to draw foundation. In our business, since we sell 1,000 nukes a year, we need to draw 5,000 frames of foundation every single year, year after year. So we are experts at drawing foundation. And so we always do it during the major honey flow right above brood. One of the tricks is don't try drawing foundation if there's a band of honey above the brood, because the bees don't want, especially if there's queen excluder above there, try to have brood right up to that queen excluder if you use a queen excluder to draw that foundation. Then the bees have all these feedback loops, written about by Schmickel and Krelsheim, how to maintain the balance between the number of nurses, the number of foragers, and the number of nectar processors, the mid-age bees. I'm not going to go into that right now. And also, brood rearing, as I said, is deprioritized. They'll fill those cells with nectar. There's no place for the queen to lay, and the amount of brood can drop precipitously right there, which is just fine because after the main flow, you don't want a bunch of hungry mouths to feed. So it's better for the colony to deprioritize brood rearing because any brood that emerges during the main honey flow, they'll be too young to actually be part of storing honey. Then we trick the colonies. There's Tim Ives out here in Indiana stacking these colonies high so they have room to store that incoming nectar. And then it comes in so fast and the humidity is high, they store this wet nectar, and then they later will then condense it down to fewer supers of stored honey. Tim, by the way, for those who are interested, I checked it out, completely surrounded as far as the bees can fly in every direction by neonic-treated corn and soybeans. And he just laughs at those who complain about that, saying, yeah, I make these monster crops of honey off those neonic-treated crops. So look at your perspective on those things. Then we get to the late summer population decline. Everything's done, dries out, no pollen, the bees go into survival mode. Attrition exceeds recruitment, so the population drops. You see the numbers going down here. And in Lloyd's data, same type of thing. We drop off. You really don't need extra mouths to feed because they want to get into this winter cluster mode. And if you look at your brood pattern, we'll have the fresh stored honey, no pollen, maybe a little nectar here, but notice all the open cells. This is because the nurses are consuming the eggs as fast as the queen lays them. They're not trying to rear any more larvae into adults right now. This indicates a protein deficit. And what we as beekeepers can do is by feeding pollen sub, you can rejuvenate, shift the age structure of the hive and get a bunch of young bees again, if that is what you want to do at that time. And now let's talk about Varroa, the other thing that you keep. It's a freebie on the web, Randy's Varroa model, Excel sheet. You can make these graphs showing the colony population, the amount of brood, the amount of mite-infested brood, and your mite wash counts. And you can enter in treatments to avoid colonies collapsing. And what happens is, notice as the mites don't really explode, but because the bee population comes down, you get a higher ratio of mites per adult bee during this downsizing phase. Typical mite buildup, if you have like a 5 or 6 month winter, you're going to go from 100 mites to 10,000 mites, about 100-fold increase here. And you can play with the mite Varroa models to see that. The mite population gets too high, you're going to start seeing Varroa sensitive hygiene of the bees trying to chew out those infested pupae. You may see bald brood. This is one of the traits that my bees are doing, my mite-resistant ones, is uncapping the brood temporarily. That disrupts the mite's reproduction. Then you may start seeing deforming virus rear its ugly head and the pupae starting to die, maybe some bees with deformed wings. And the reason is if we look at mites per 100 bees and you look at the prevalence of highly infected workers over time, as the mite infestation rate increases, the viruses just take off. Deforming virus here, cashmere bee virus family right here. You want to keep your mite level down to around here. We, like in our operation, don't like it to ever get above about 6 mites in an alcohol wash of a half cup of bees. Or we actually do Dawn detergent washes these days. You also reach a tip point if over about a third of the pupae are infested by a varroa mite, and then the colonies typically die shortly after that. And at that point, you start to see bees unable to emerge from the cells, maybe deformed wings, parasitic mite syndrome, where the colony looks very, very sick, and a close-up of parasitic mite syndrome, or PMS. And then finally, you see all these fecal deposits from varroa mites at the tops of the cells, indicating it was varroa that caused the colony mortality. Close-up of the fecal deposits here. The problem is when those colonies get to those high mite levels prior to collapse, there are bees drifting all the time. Here's a mite on one bee, another mite, and there's more mites underneath these bees. There's this constant diffusion of mites throughout the landscape. Plenty of them up to a half mile away. I've tested this by marking bees, independent of any robbing, just simply by bees drifting all the time. And with them, they take these mites that drift into other hives. This is a very real thing, this mite immigration from the high mite hives. As far as treating for varroa, the most common method plan for treating by new beekeepers is called wishful thinking. And I'll tell you right now, it has never, ever worked. Okay, typically you're going to need about 3 reductions of the mites throughout the year to really keep your colonies healthy. And you want to get them down to close to 0 mite wash counts going into the winter. In our operation, we split our colonies in the spring to create an induced breed break, hit them with an oxalic acid dribble. While the honey super's on, we'll hit them with a formic treatment to knock back the mites back a bit. Then a thymol treatment in August. Feed protein to build up our bees for winter, and then an oxalic acid dribble in late November or December. And this has worked for us for years very successfully. We're also in the middle of a selective breeding program for natural mite resistance. These are alcohol wash counts over the course of the season from July to November here. And we only produce queens every year from those colonies that control the mites by themselves. Okay, so let's get to autumn now. We start getting fall pollen coming in, and now the bees may store surplus pollen beneath the brood nest for winter stores. You want to fatten these last bees emerging up with a lot of pollen. And here's, you can see with Lloyd's data, these bees are all going to die off, the older bees up here. And it's just this last rounds of brood right here, the September, October bees emerging that are going to become your winter cluster. And Heather Madeline Gardotis did this very nice paper on this, showing this is your proportion of surviving bees, starting with 100% with these emergence dates. Notice the bees that emerge in late summer die off very quickly. Then you get into, this is up north here, those that start to emerge in September and October. They're long-lived winter bees until they start rearing brood here in March or April. And then bingo, they go right back to that short longevity again. So these winter bees, the ones that emerge in late season, are the most important for wintering. So we successful beekeepers, first they control their mites in August, and then they feed in September with protein, unless you have a natural pollen flows. And on the East Coast, you don't need to feed at all because you got plenty of pollen. Out in the West, you don't. So we then feed pollen to initiate and sustain that late season brood rearing of mite-free, virus-free bees that will become the winter cluster. And if the colonies don't consume that pollen sub, we just write them off. We don't even waste time looking at them anymore. They got an issue. And then when we get a first frost and there's no more pollen coming in, brood rearing is reduced to essentially nothing for a while. And at this point, when these bees emerge, they don't smell any brood. They don't have any other emerging bees to push them into going into foraging. So they remain in this nurse physiology, or these diutinous long-lived bees, where they just are survivors. They build their fat bodies heavy Whereas foragers deplete their fat bodies, and this is what varroa feeds on, is these fat bodies. So nurses or winter bees look like this. The other thing they do is going into the winter, they load their guts full of pollen. Here's a sample of 50 bees in November, with every single bee with a gut load of pollen. During the summer, you have a much lower proportion that ever have a gut load of pollen. In the fall, turnover, where you just get this sudden drop. Around us is about November, and half the population of the hive just flies off to die from old age. Then you get the winter cluster formed. And it looks just like a ball of bees, but if you give them a puff of smoke here, you'll see they'll separate out. The bees that are warm and can move compared to the bees that can't move will look like this. So this is the insulating shell, and then the tropical environment right in the middle. And from a side view, it would look like this. This would be a layer of bees with their heads pointing in the insulating shell, pumping their hemolymph from their tail end towards the head end, pushing that countercurrent, pushing the heat back in. And then the bees in the middle, you have these heat generating bees, which will raise the temperature. This temperature stays around 50 to 65 degrees Fahrenheit out here. They won't let it drop below 50 degrees. And then here it's much higher. It's an airtight cluster. So the carbon dioxide level can go way up, the oxygen level goes down, and they tend to open up the bottom and ventilate moisture out. That moisture then, the heat of vaporization, can then be captured by the colony and help them. So I really have a hard time with beekeepers recommending top ventilation, because it's just contrary to everything that bees want to do. They like to bottom ventilate out. But what you can do is put insulation over the top of the hive. So you can see the heat loss here. That means you're going to have condensation on the inside dripping down. So put a thick layer of dry, not any insulation that can get wet. You don't want to absorb moisture because it loses insulation value, but dry insulation above the top of the hive. So a piece of styrofoam or something works very, very well. And now we have these long-lived bees, which will live all through the winter right here. And scant protein reserve, you don't see any stored pollen. Bees are only able to rear maybe one generation of brood without bee bread or pollen coming in. And then we start the initiation of brood rearing. Now, the bees may initiate brood rearing during the winter, and this seems to be maybe for water balance, that if they can't fly out to defecate, what they'll do by rearing brood, they can use that water then, absorb it, and use it to produce jelly to use up that water by feeding brood. So this is an area that needs more research. And the beekeeper can stimulate the colonies then for late winter brood rearing. We do this a lot prior to almond pollination. Here's my friend Keith Jarrett, who during the winter feeds huge amounts of pollen sub, and in January he has these monster colonies he can actually sell bees from to other beekeepers. And then we're back to the first tree pollens coming back in. The cycle begins again, and we have reached the end of the year. Again, keep young queens, good nutrition, control varroa, minimize toxins, and be proactive rather than reactive. Lots more information at Scientific Beekeeping. And that's the end of it, guys. I hope you're doing well. I hope my voice holds up and I'll be able to maybe answer some questions. Okay, thank you.